My name is Dr Joyce Harper and I work at University College London in the Institute for Women's Health as part of the UCL Centre for PGD team. And I've worked in IVF for just over 25 years and in PGD for just over 20 years. And today I'd like to talk to you about reproductive genetics, pre-pregnancy and pre-implantation genetic testing. My talk today, I'm going to cover what is PGD and PGS, how we do these procedures, something about the ESHRE world data and our UCL data, the disadvantages and advantages of these procedures, pre-pregnancy genetic testing, the future of these procedures, and some of the ethical concerns that we have. So what is PGD? Well, some people already may know what PGD is, but I just want to briefly set the scene so you can see where this is different than PGS. So PGS is a procedure that we do for fertile patients that are at risk of transmitting a particular genetic or chromosomal disease to their children. So these couples, for them, their main reproductive choice would be maybe to go through prenatal diagnosis. So this is a test where we do an amniocentesis or a chorionic villus sampling on an already established pregnancy. And we do the test and then look for the disease that the patients are carrying. And we can tell the patients whether their fetus is carrying the disease or not. And if the fetus is carrying the disease, the couple then can make some informed choices about what they want to do with that pregnancy. But that's a very hard situation for them to be in. For many people, they want to start the pregnancy knowing that their child is free from the disease. So that's where we do PGD. So for PGD, we do almost exactly the same as prenatal diagnosis, but we do the test on the pre-implantation embryo, so the embryo before it's implanted. So to do this, the couple have to go through IVF procedures so that we can generate the embryos in our lab, we can do a biopsy to remove some of the cells, and then we can do our genetic testing on the embryo. So the aims of PGD are to identify genetically and chromosomally normal embryos with the view that we transfer these normal embryos to have a healthy child for that couple. At the current times, we're just doing PGD for the disorder the couple are carrying, and the current techniques are relatively uncontroversial. But as you'll see by the end of my talk, there's a number of new technology that's coming in that's allowing us to test for a lot more than just the disease the couple are carrying. So the indications for PGD, well, any single gene disorder can be done by PGD, so these can normally fall under recessive, dominant or X-linked, and this includes the triplet repeat disorders and also late onset disorders. So this will be things like cystic fibrosis, beta thalassemia, haemophilia. These are diseases we can diagnose by PGD. The couple may be carrying a chromosome abnormality, such as a Robertsonian or reciprocal translocation, or another type of chromosome abnormality. And in these situations, the couples may experience recurrent miscarriage because these embryos are not viable with life. And we can also do PGD for these couples. There are some diseases that are caused by a single gene, which is carried on the X chromosome, and these are called X-linked diseases. And the ideal way to do this is a specific diagnosis of that particular X-linked disease. So we can differentiate affected embryos, carrier embryos, and totally normal embryos. But we could just do sexing and transfer the female embryos, because these are diseases that are normally only manifest themselves in a male offspring. Now, in the UK, all the IVF and PGD that we do is governed by the Human Fertilisation and Embryology Authority. And if you visit their website, they have a list of the diseases that they have licensed for PGD. So if the disease falls on their website, then we can do PGD for that. If not, then we have to apply for a specific licence. So all the PGD in the UK is licensed by the HFEA. So that's quite clear now. So what is pre-implantation genetic screening? Well, this is using the same technology that we use for PGD, but this is now as an adjunct to IVF procedures for fertile or infertile or subfertile couples. So these are patients that are going through IVF anyway because of infertility, and the aim here is to select the embryos to transfer on the basis of their chromosome complement to try and increase the pregnancy rate for certain groups of patients. So really this is using the IVF and PGD technology to help select the best embryo for transfer. So for this to work, the aim of this procedure is to try and improve the delivery rates. So what we would need to do is have evidence that this procedure is actually doing that. And we can do that with something called a randomized controlled trial. A randomized controlled trial is a scientific experiment where you'd have some people would have the treatment and some people wouldn't have the treatment. And you would see if the treatment group had a better effect from having the treatment than the control group. So I'll come back to some of those RCTs later, but PGDS has been a controversial technique, and as part of the ESHRE consortium, we've written a couple of statements on this, and there have been some by many others. But I'm going to come back to that after I've explained how we actually do PGD and PGS. So the indications for PGS, well, advanced maternal age. We know that as women get older, they're at less chance of getting pregnant, and one of the main causes of this is chromosome abnormalities in their eggs. So once a woman gets sort of 35, 37, the level of chromosome abnormalities in her eggs starts to increase quite significantly, and certainly over 40 it becomes very significant. So we could do PGS, look at the chromosomes, and transfer embryos that are chromosomally normal. Also, patients with recurrent IVF failure, so 2 or more failed IVF attempts. This is a very tricky group of patients because if they're having failed treatment, there could be a number of reasons why they're not conceiving. But chromosomes in the embryos may be one of them. Recurrent miscarriage, where they're having a recurrent miscarriage, but their karyotypes are normal. So they're not known to be carrying a translocation, which would lead to recurrent miscarriage. So again, there are many reasons why recurrent miscarriage may happen. Chromosomes in the embryos may be one. And also severe male factor. So let's look at the stages of PGD and PGS. Well, there's 2 stages. Once the embryos are in our lab, we perform the embryo biopsy to remove some of the tissue that we can then examine, and then we're going to do our diagnosis from 1 to 5 of the cells. So there are 3 main methods of embryo biopsy. Polar body biopsy, which is shown in the top diagram, and this is where we take the first and the second polar body from the egg and the early zygote, and use that for our genetic analysis, and that will tell us about the chromosomes from the mother. Cleavage stage biopsy is the most commonly used method at the moment, and this is where we take normally 1 cell from the cleavage stage embryo, which is around day 3 of development, and the embryo will be about 8 cells by this stage. Blastocyst biopsy is becoming a lot more popular now. The embryo reaches the blastocyst stage around about day 5 or 6 of development, and at this stage we can remove some of the trovectoderm cells. These are the cells that you'll see here that are being aspirated, and these cells are cells that will go on to make the placenta. So we can aspirate those cells and then find out what's happening with the chromosomes or genes in the embryo. So once we've done our biopsy, we put the embryos back into the incubator and wait while we do the diagnosis. And the diagnosis can be done by 3 methods depending on the type of abnormality we're looking at. So the PCR technique is used to look at mutations. This includes single gene defects. And so what we do with PCR is we amplify a particular region of the DNA thousands of times, and then with these copies of the DNA, we can do specific tests to identify whether the abnormality is present or absent. We have used fish, fluorescent in situ hybridization, to look at chromosomes. This is a picture of a fish on a blastomere here. And we can use these fluorescent probes for specific chromosomes, and they can tell us whether those chromosomes are present or absent in the embryo. So this has been done a lot, for example, embryo sexing, where we can use probes for the X and Y chromosome, and that will tell us if the embryo is male or female. But fish is almost being totally replaced now by a procedure called array CGH, array comparative genomic hybridization. And arrays are a very elegant way to look at all of the chromosomes. So with fish, we can only look at a few of the chromosomes. With arrays CGH, we can look at all of the chromosomes within an embryo. And it's also something called single nucleotide polymorphic arrays, or SNP arrays. And with them, you can look at the chromosomes, but you can also look at the genes. So these are new techniques that we're now using in PGD. So this is an array CGH of a single cell from a trisomy 10 cell line. And we use this to validate the arrays in our own laboratory. And this is chromosome 1 here. These shaded bars are the different chromosomes. So chromosome 1, 2, 3, all the way here to X and Y. So anything above the line means there's an extra copy of the chromosome there, and anything below the line means there's a missing copy of that chromosome. So here you can quite easily see that there's an extra copy of chromosome 10. So this gives us indicates it's a trisomy 10 cell, and this was indeed some validation work we did with the trisomy 10 cell line. Now, I couldn't talk about PGD without talking about chromosomal mosaicism, and this is unfortunately present at all stages of pre-implantation development. Work that Joy Delhanty and I did back in the very early 90s, we showed that at least half of cleavage stage embryos were mosaic. So this is where you have some normal cells and some abnormal cells, or even two different cell lines of abnormal cells. But the most common abnormality we see is what we call diploid mosaicism, where some of the cells are normal, but some have an abnormality. And many other people, Santiago Monet and others, have now done work that validated our findings back in the early 90s. So the problem for PGD and PGS is that we're biopsying a cell from the embryo, and if the cells from the embryo have a different chromosome makeup, then the cell we take out is not necessarily representative of the rest of the embryo. So this mosaicism certainly leads to problems when we're identifying chromosome abnormalities. In blastocysts, though the later stages, around day 5 or 6 of development, about a third of the embryos have mosaicism. And Dagan Wells and his team have done a lot of work on blastocysts at this stage and had a look at the level of mosaicism and feel it's about 33%. So this really is a problem for PGD and PGS. But if we're aware of the problem, we call it the biological problem, we're at least fully informed about the limitations of the procedure. So let's go back to have a look at PGS. So as I said, these randomized controlled trials, there are now more than 11 randomized controlled trials that show that there's no benefit of using PGS. These have all been done using FISH, and 10 of them were done using cleavage stage biopsy. And the conclusion back in 2010 was that cleavage stage biopsy and FISH may not be the right time and the right procedure to use for PGS. And so the ESHRE PGD consortium wrote a position statement in human reproduction, what next for PGS, a position statement from the ESHRE PGD consortium steering committee. And in that statement, we said that at the current time, we shouldn't pursue FISH and cleavage stage biopsy, but we should look at the new technology, the arrays that were coming out, so we could look at all of the chromosomes, and also look at polar body biopsy and trophectoderm biopsy as alternative stages. So the ESHRE group have been very active in this area, and we set up an ESHRE PGS task force back in 2007, and we felt we had to learn from the PGS mistake. There's been many thousands of cycles that have been done, and all the scientific data shows that this technique is not of benefit. And actually some of the RCTs showed that the PGS treatment group had a lower pregnancy rate than the control. So we were looking at the new technology that was coming in at 2007, and that's when the arrays first came into the PGD arena. And what we felt was the most important question at that time was, is this method validated? And if the arrays were validated and were found to be accurate and useful for PGD and PGS, we would have to go on to look at a randomized controlled trial to show that it's of benefit to IVF patients. And this would have to be an increase in delivery rate. Some of the early studies were looking at pregnancy rate and ongoing pregnancy rate, but really the delivery rate's the important thing. So for PGS, we need to evaluate polar body and trophectoderm biopsy. So the work of the PGS task force, all the hard work was done by Christina Magli's lab in Sismo in Bologna, and Marcus Montag's lab in Bonn in Germany. And their teams did a very large study, very well-designed study, to validate the use of arrays on polar bodies. And 2 papers were published from this pilot work. And I felt very strongly, since I've been working in IVF, that we're bringing a lot of technology for IVF patients that hasn't been validated and hasn't shown by an RCT to be useful. So I felt it important to write a paper on when and how should new technology be introduced into the IVF lab. So we published this in 2011 with some of the key embryologists and andrologists in Europe. And then one of my master's students and myself, Rachel Brown, we wrote a paper looking at the safety and future of these procedures. So for new technology in IVF, in any aspect of IVF, we really need to show that it's a valid procedure and that it is of benefit to the patient. And the best way is to do these RCTs. So let's look at the ESHRE PGD consortium. A lot of information about the consortium can be found on the ESHRE website. And every year within the ESHRE PGD consortium, we have a growing number of centres that are registered with the consortium, and it's very much an international membership. So we have centres from the US, from the Middle East, Australia, really all around the world. And some of these centres send in very detailed data about their PGD and PGS cycles, so we can find a lot about what happens in PGD around the world. And we've been collecting this data since 1997, so we do an annual report of the data collection and also look at the cumulative data. So in 2012, we thought it would be a good idea to publish the first 10 years of our data collection, and this paper is freely downloadable from the ESHRE website. So here you can see these green and red dots. These are our full and associate members, and you can see how we really do scan the globe. The consortium's done a lot of work. Besides data collection, we've written guidelines. We had first guidelines out a few years ago, but last year we updated the guidelines and wrote 4 guidelines on specific aspects of PGD and PGS. Now, accreditation and external quality assessment are really key for any diagnostic lab. And our lab at UCL was first accredited about 5 years ago. And the consortium felt it very important to help labs in this area. So we've been involved in training and also helped set up external quality assessment schemes. And this is now being headed by Siobhan Sen-Gupta, who's my colleague who works with me at UCL. So some of us, including myself and Siobhan, when we went through the accreditation process, we thought it'd be useful to write a help guide for others that were going through this process. So we wrote a paper back in 2010 on accreditation of the PGD laboratory. And we took the regulations, the ISO that we're involved with for PGD and PGS, and went through every stage and how the PGD lab can go through the accreditation process to help other labs. So let's look at a bit of the data. As I've said, this is the review that we've written of 10 years of data collection. So our first data collection was in 1997 when we first started, and we separate the data into PGS, social sexing, sexing for X-linked diseases, but this can also be done by specific diagnosis, chromosome abnormalities, and monogenic or single gene defects. So we can see obviously the number of cycles performed has grown over the years. But another important point is that PGS accounts for more than all the other types of PGD put together. And even with the RCTs coming out, PGS is still a very huge area of our field. At UCL Centre for PGD, we did our first cases in 1991. We collaborated at that time with the Hammersmith Hospital, with Alan Handyside and Robert Winston. And Joy Delhanty and Darren Griffin were the first people that did those cases back in 1991. We're one of the few academic PGD centres. We're obviously part of University College London, and we've been accredited since 2009. And we have many PGD firsts, both national and international, and please visit our website for further information. So this is the list of diseases that we've done PGD for, and as I've said, these will all also be listed on the HFEA website. And we've broken up the results into different batches, but our data from 2000 to 2011, we've had over 1,000 referrals, and we've done 483 egg collections and delivered 110 babies. So our live birth rate per egg collection at that time is about 26%, and we've had 124 babies born, some of them were obviously twins. And just the results for 2012, we've done a little bit of PGS, but we've mainly done PGD for single gene disorders and translocations. So we could do PGD for single gene disorders, including de novo mutations in male and female partners. Chromosome abnormalities are mainly now by erased CGH. And the work that we do with the Centre for Reproductive and Genetic Health, who we work with at the moment, we do polar bodies, single blastomeres, and trivectoderm biopsy. And the CRGH was awarded NHS contract to do 60 patients for PGD in 2013. So we're working with them to deliver those patients. Now, let's look at the advantages and disadvantages of PGD and PGS. Well, the patients have to go through IVF, and that is a very tall order, and it's very hard for fertile patients to go through the quite involved stages of IVF. It's also very costly. Some of our patients are funded, but some are not fortunate enough to get funding, and the cost of a PGD cycle is very expensive. We do have cases where all of the embryos have been affected, so we can't transfer any embryos. That's obviously very, very difficult for the patients. And we are making a diagnosis from just a few cells, which is a very tall order. Any other genetic test, you'd have buckets of cells and buckets of DNA to do your genetic tests. So unfortunately, there have been a few misdiagnoses. It's a very, very small percentage of the PGD and PGS cycles, but there have been those even in some of the best IVF units in the world. So we have to be very careful about how we do these tests. But because of the biological problems, such as mosaicism, sometimes we can't be 100% accurate because it's just out of our control. Now, PGD, I often say the success rate is lower than IVF. In a normal IVF cycle, even though you've got infertile couples, you are generating embryos in vitro, and you're just nurturing those embryos. You're not doing anything else to them, and you're choosing the very best embryos to transfer. In PGD, we have to remove some of the cells, so we're doing quite an invasive procedure, and then we're transferring the embryos on its genetic status. So we might not necessarily transfer the best looking embryo, the embryo with the best morphology, but we'll transfer the embryo that's free from the disease. So those that have an effect of lowering our pregnancy rate compared to IVF patients. And the CRGH has a pregnancy rate for IVF in well excess of 40%. So you can see for PGD that is lower. In PGS, the aim of using this technology is to improve the delivery rates. So we don't want any effect that's going to reduce it. But we have already seen that in some of the randomized controlled trials. So I think one of the problems has been that the effect of the biopsy and the invasiveness of the biopsy to the embryo is outweighing the advantage of transferring embryos that are chromosomally normal. But I think also the mosaicism has a problem in sometimes we're not getting the true picture of the embryo because we've taken out one of the cells that are not reflective of the rest of the embryo. So the advantages of PGD and PGS, well, the main advantage of PGD is that in the majority of cases, you will be starting the pregnancy knowing the embryo is free from the disease. So for PGD, it avoids the need for termination of pregnancy. For PGS, we have to have the randomized controlled trials to show that this is improving our delivery rates. For some people, they may use this as a closure after many IVF cycles. We've had patients that come through for multiple IVF cycles, and it's very hard for them to stop. The financial issue is obviously a big problem. So they may decide to go through PGS to find out if any of their embryos are chromosomally normal. And if they are not, then that may help them stop going through any further IVF cycles. But the data on repeated IVF failure and recurrent miscarriage are not really available. There's recently this year been an RCT on repeated IVF failure that did not show any significant results. So we have to really see how it develops in the future. So where are we in the future of genetic testing? Well, I think something that's very interesting now, but also a little bit scary, is the direct-to-consumer testing. So any of us can go on 23andMe on their website, there's the web link there, or on Decode&Me, and you can follow the instructions there, and for a couple of $100, you can send them some DNA, some of your DNA, and they actually do a SNP array and find out a lot of information about certain diseases that you may be prone to. And also, if you do this as a couple, they'll tell you some information about the possible genetic consequences of your resulting child. So I think in a way that's a little bit scary, finding out all this genetic information about yourselves, especially if you don't have genetic counselling, if something actually gets shown up. You may find out you've got a late onset disease for which there's no cure, and once you've got that information, you can't take it away. So I think we have to be quite careful about how we use this information. But something we're moving to in the future, it was James Watson back in 2007, so it was Watson and Crick that really decoded the structure of DNA. And in 2007, for $1.5 million, his genome was sequenced. So almost every base of his genome was determined, so we could get his full genetic code. Now, things move very quickly in the development of this new technology, and here's something from a Illumina website, and I think that says that it's about $9,500, and that was a few years ago. So now it's getting even cheaper. So this is the most recent data from another website where, again, you can send in your sample and get your own genome sequenced. So in some ways this is exciting, but in some ways, as I said, it's scary because you need to have counselling. If something gets shown up in these tests, as it will for some people, the patients do need to have information about what that really means to them. There's also a number of sites that are opening up that are doing pre-pregnancy genetic screening. So this is just two of them. This is Counsel and Pathway.com, Pathway Genomics. And they offer tests to couples that are thinking of starting a family. So these are, again, using SNP arrays at the moment, and they'll test for about 100 of the common mutations, such as cystic fibrosis, beta thalassemia, and they'll give you an idea of whether, as a couple, you're compatible or not genetically. And if you are carrying any recessive or dominant diseases, then again you would definitely need counselling to find out what that meant for your children. But then you could have the necessary genetic tests. So these things are coming. This technology is here. I just think we have to be careful and use this slowly and carefully with the correct counselling. So the future of PGD, we've got this new technology that's allowing us the diagnosis of more disorders. We've got SNP arrays that are telling us a lot more information about the embryos, and sequencing is here already. And already we can do non-invasive prenatal diagnosis when there's a sample of the maternal blood that's taken, and that blood can be sequenced, and you can take away the sequence of the mother, and you will get left with the sequence of the child. And this has already been done for the main trisomies, such as trisomy 21. So that's amazing that that technology is coming in. It's a bit slow to come in at the moment. We have to, again, be careful and make sure that we really have accurate data that's going to be sure to give us the correct diagnosis. We're moving more towards blastocyst biopsy, and I haven't mentioned yet, but there's a relatively new method in IVF called vitrification, and vitrification is a way of cryopreserving or freezing embryos, and it's very successful to be done on PGD or PGS embryos after the biopsy. So we can freeze all the embryos and do a number of genetic tests, or freeze the normal embryos and keep those for a future cycle. So that's another exciting advance in this field. So just to finish off, the ethical dilemmas in PGD. There's something called HLA matching or save your sibling, and this is where we have an existing child who's very ill, who's got a life-threatening condition. It may be, it's always a blood disorder, so something like beta thalassemia or sickle cell, or also something that may not be inherited, such as leukemia. So to save that existing child, there's a number of options for them, but one main option is that they have a bone marrow transplant from their matched sibling. But if there is no matched sibling, then there's, again, some options, but one of the options would be for the couple to go through PGD, and we would match the embryos that were generated to see if any were a HLA match, a tissue match for the existing child. If they are a tissue match, then the embryos can be transferred, and at delivery we can use the cord blood to hopefully cure the existing child, and that would be a fantastic situation. It's a little bit more complicated than that. If you watch the movie My Sister's Keeper, that's where a child has leukemia and they've gone through exactly that procedure, and you'll see in that film there's lots of different things that we have to think about. Sex selection, it is illegal in the EU, but with PGS and with some PGD, you would determine the sex. So I think it's probably being done a lot more than we think, and certainly in the US it's legal. So there's always a mixed view about sex selection. Some people feel it's fine, and some people feel it's not. So that's a very personal thing. I think that's something we need to continue to debate. But non-life-threatening, who decides? So the SNP arrays that are around at the moment, you can check your diabetes risk and other non-life-threatening risks. And where do we really decide if a genetic disease is life-threatening or serious, whether we should be allowed to do these procedures or not? So there's really a lot of ethical discussions that we are always involved with to try and think about these issues. We definitely don't decide anything because it's a very individual view. And SNP arrays and sequencing, we can test for everything in one go. So another thing to go and look at is Gattaca. You can go onto YouTube and look at the trailer for Gattaca or watch the film. And this was a film from quite a few years ago now, but it says in the not too distant future, basically everyone reproduces by PGD, and we sequence the genome and we only transfer embryos that are going to be super healthy, so that it would keep medical costs and education, everything being much more efficient because we'd be dealing with healthy people. So it's a very scary situation, but really it's a situation we're almost at now. And within ESHRE and other groups, we discuss all of these issues quite regularly. And a few years ago, we had a meeting in Maastricht with some of the ethicists and some of the PGD people, and we wrote this paper, Dynamics and Ethics of Comprehensive Pre-implantation Genetic Testing, a review of the challenges. So this looks at the ethics of the new technology. And we've just recently submitted a paper, again from an ESHRE and the European Society of Human Genetics collaboration. And we wrote this very lengthy paper on current issues in medically assisted reproduction and genetics in Europe. So this is our team at UCL Centre for PGD and at the CRGH. And our team is headed by Joy Jalhanty, myself, and Siobhan Sen-Gupta. And these are some of our PGD staff and some of our PhD students. And this is just some of the members of the Centre for Reproductive and Genetic Health. The medical director is Paul Sahal, Karen Doy is our genetic nurse specialist, and Alpesh Doshi is the head of our IVF lab. And I've written 2 books on PGD, one more than 10 years ago, and then I updated it in more recently, a few years ago, and they're both called Pre-implantation Genetic Diagnosis. Thank you.